lapel.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Naphthoquinones and their derivatives.
And specifically their role or potential role in fighting cancer.
Yeah, and it's crucial work. Cancer is still such a massive global health issue. Finding new treatments, new approaches is absolutely vital.
And these articles focus on naphthoquinones, these other natural compounds.
They are often naturally occurring, yes, found in various places in nature, and they have this really a broad range of biological activities. But the focus here, the really exciting part for this discussion, is their anti-cancer effect, their anti-neoplastic activity.
Okay, naphthoquinones. It definitely sounds technical. So what are they fundamentally? Why are they catching researchers' eyes in the cancer field? What's the basic idea?
Okay, so think of quinones as a big family of chemical compounds. Naphthoquinones are just one type within that family, defined by a specific structure, a naphthalenic ring. Quinones generally, well, they can be made synthetically or found in nature, and they have roles in metabolic cycles.
Sort of fundamental biological processes.
Exactly, and within quinones, you've got a few main groups: anthraquinones, benzoquinones, and our focus, naphthoquinones. The key thing for us is that naphthoquinones are actually pretty common. You find them in microbes, plants, even some animals.
Right.
And some of these naturally occurring ones have shown real promise against cancer.
Okay, so they're out there in nature. Are there specific ones that are already known to be active? Kind of the front runners.
Oh, definitely. There are several natural ones that have shown significant bioactivity. Names like plumigin, lawsone, lapicol, juglone is another one.
Yeah.
Alapechum. And these aren't just sitting there, they do things. They have pharmacological properties. We're talking anti-cancer, yes, but also antimicrobial, anti-inflammatory, even anti-allergic and anti-platelet effects. A really wide range.
Wow, that's quite a list of activities. And I guess scientists aren't just looking at them as they are found in nature. You mentioned derivatives earlier. They're tweaking them.
That's exactly it. You can take the basic naphthoquinone structure, the 1,4-naphthoquinone core, and you can modify it, add different chemical groups.
Little attachments.
Precisely. And doing that changes its properties. Things like, how easily it accepts or gives up electrons, its acidity, and critically, how it interacts with biological targets, like proteins in a cancer cell.
So it's like molecular engineering almost. Building a better version.
In a way, yes. It opens up this huge potential to create these modified naphthoquinones, these derivatives, that might be even better at targeting and killing cancer cells. And interestingly, even the substituents, the parts added on, can have their own beneficial activities: anti-allergenic, apoptotic, which means causing cell death.
Okay.
Antifungal, antibiotic, antiviral, antiplatelet, you name it, anti-inflammatory, radical scavengers. It's quite versatile.
So you're essentially creating a toolkit to build more precise weapons against cancer. And the research suggests these are already showing promise against specific cancer types.
Yes, definitely. Compounds with this naphthoquinone structure have shown benefits against liver cancer, breast cancer, human cervical carcinoma, gastric cancer, a range of them.
How do they actually kill the cancer cells?
A key mechanism seems to be inducing something called autophagy.
Autophagy. Sounds like self-eating.
That's basically what it is. Think of it as the cell's internal cleanup and recycling system, but it can also trigger programmed cell death. Naphthoquinones seem able to push cancer cells towards this self-destruct pathway.
And this involves specific signaling pathways.
Right. Autophagy is controlled by complex pathways inside the cell, like the PI3K and MTOR pathways. These are like master switches for cell growth and survival. Naphthoquinones appear to mess with these switches in cancer cells, pushing them towards death.
Okay, so we've got a picture of what they are. Let's get into the nitty-gritty now. How exactly do they attack cancer cells? What are the main strategies?
Right. This is where it gets really interesting mechanistically. Their effectiveness, their deadly impact on cancer cells, seems to come from several different angles.
Not just one trick, then.
No, not at all. One major way is by inhibiting electron transporters, often in the mitochondria, the cell's power plants. This messes up energy production.
Cutting off the power supply.
Essentially, yes.
Yeah.
They can also uncouple oxidative phosphorylation, which also disrupts energy generation. Another big one is creating reactive oxygen species, ROS.
ROS. We hear about those a lot, like damaging free radicals.
Exactly. Think of them as tiny, highly reactive sparks inside the cell. They can damage crucial things like DNA, proteins, causing chaos within the cancer cell.
Okay.
And then they can also directly interact with proteins. They can form adducts, basically sticking onto proteins, especially those with specific sulfur groups, the sulfhydryl or aromatase H groups, which are common in important enzymes. This can shut down those enzymes.
Wow. So disrupting energy, causing damage with ROS, and disabling key proteins. That sounds like a multi-pronged attack.
Yeah.
Harder for the cancer cell to fight back.
That's the idea. Hitting multiple targets makes it potentially harder for resistance to develop, which is a huge issue with many current cancer drugs.
Makes sense.
Yeah.
The research also mentioned inhibiting something called topoisomerase 2. What's that about?
Yes, topoisomerase 2. That's another important target. It's an enzyme that's absolutely crucial for DNA replication and cell division.
Which cancer cells do a lot of.
Exactly. They're dividing rapidly, so they rely heavily on topoisomerase 2. By inhibiting this enzyme, naphtha-decanase can basically stop cancer cells from multiplying. And one specific naphtha-quinone, lapachone, which we mentioned earlier, lab studies, in vitro studies, confirmed it's an irreversible inhibitor of topoisomerase 2. It basically shuts it down permanently.
Okay, that's pretty potent. And what about the connection to P53? That's a famous tumor suppressor gene.
Right. Absolutely. P53 is critical. It's often called the guardian of the genome. It responds to DNA damage and tries to stop cells from becoming cancerous. And it seems naphtha-quinone activity is heavily linked to P53.
How so?
Well, studies show that when naphtha-quinones cause DNA damage in cancer cells, they can trigger P53 activation.
The guardian wakes up.
Kind of. And this P53 activation can then lead to other things, like reducing the levels of a protein called CDK4, which pushes the cell cycle forward. Ultimately, it can trigger apoptosis, that programmed cell death pathway in the cancer cells.
So they're recruiting the cell's own defenses against itself.
In a way, yes. For example, another naphtha-quinone, chiconin, has been seen to increase levels of BAX, that's a pro-death protein, and decrease BCL2, which is anti-death. So it tips the balance towards cell destruction, mediated partly through this P53 response.
Fascinating. There was also mention of MEL-T1 inhibition. Is that another general mechanism?
That one's a bit more specific. It relates mainly to certain types of lymphoma, cancers of the immune system's B cells. Beta-lapachone, that potent one again, was found to be a strong inhibitor of this MEL-T1 protein. MEL-T1 is important for the growth of these specific lymphoma cells. So by blocking MEL-T1, beta-lapachone shows real promise for inhibiting those particular cancers, at least in lab studies.
Got it. Okay, so we have a good handle on the how. Let's talk specifics. You mentioned juglone earlier from walnut trees. What's its story with cancer?
Right, juglone. It's naturally present in walnuts. We know it has antibacterial, antiviral, antifungal properties too. But in cancer, it's shown some anti-metastatic effects on pancreatic cancer cells.
Meaning it stops the cancer spreading.
It seems to help prevent the spread, yes. Studies suggest it triggers dose-dependent apoptosis, more juglone, more cell death by increasing those ROS levels we talked about, messing with the mitochondria's membrane potential, and affecting key apoptosis genes like BAX and caspases, while reducing BCL2.
Sounds promising for pancreatic cancer, which is notoriously difficult.
It is, but it's important to stress, these anti-metastatic studies are still quite limited, mostly lab-based right now.
Understood. Early days.
Juglone has also shown some action in endometrial cancer, incurring a type of cell death that depends on iron, called ferroptosis, and also blocking the migration or movement of those cancer cells.
Okay. Moving to prostate cancer. The research noted progress, but also this big challenge of drug resistance.
Yeah.
Could naphtha-quinones help there?
Yeah, prostate cancer treatment has come a long way. Death rates are down, but it's still a major killer. And resistance to newer drugs like abiraterone or enzalutamide is a real problem.
So new options are needed.
Definitely. While the articles we looked at didn't pinpoint a specific naphtha-quinone ready for prostate cancer clinics today, the general mechanisms hitting multiple targets generating ROS suggest they could be valuable. And actually, one review did mention newer synthetic naphtha-quinones being tested against prostate cancer cell lines, so it's an active area.
Right, and oral cancer, often linked to HPV, and treatments can be tough. Any hope from naphtha-quinones there?
Well, the sources highlight that many powerful anti-cancer drugs do come from natural materials originally. Oral cancer often has poor survival rates and treatment side effects are significant.
Yeah.
So while no specific naphthoquinone was named as a current oral cancer treatment in these papers, the general anti-cancer properties make this class of compounds a very interesting pool to draw from for developing potentially less toxic future therapies.
Makes sense. What about stomach and esophageal cancer? Often aggressive. There was mention of a specific pigment extract.
Yes, that was interesting. A study looked at an extract called naphthoquinone pigment Ella3 from a plant, Arnebia euchroma.
Okay.
And this extract reduced the growth of stomach and esophageal cancer cell lines in the lab. The encouraging part was that it seemed effective at doses that didn't harm normal human cells nearby.
That selectivity is key.
It really is. It also significantly cut down the cancer cell's ability to form colonies, which is crucial for cancer growth and spread. So, definite potential there for those tough cancers.
And briefly, skin and cervical cancer.
For skin cancer, plumbagin, isolated from Plumbago zeylanica, showed it could inhibit the development of squamous cell carcinomas caused by UV radiation.
So, preventing skin cancer development after sun damage.
That's the implication from those studies, yes. And for cervical cancer, plumbagin again pops up. It induces those reactive oxygen species, mediating apoptosis in human cervical cancer cells.
Okay, it's really clear that these compounds, both natural and modified, have broad potential. Let's shift to the recent research. What's happening right now? What are scientists cooking up in the lab that's exciting?
Yeah, this is where we see real progress.
Yeah.
One of the articles detailed the synthesis, the creation and testing of a whole new library of naphthoquinone derivatives.
Designed specifically for anti-cancer activity.
Exactly. And several of these new compounds, they had specific labels like 5I, 5I, 5O, 5Q, 5R5s, 5T, and 5V showed superior cytotoxic activity. They were better at killing the cancer cells they tested compared to existing benchmarks.
Wow. And I think I read something about better safety profiles too, compared to standard chemo.
Yes, that was a critical finding. All the studied compounds showed a higher selectivity index, or SI. That means they were preferentially toxic to cancer cells over normal cells.
More targeted.
More targeted, exactly. And they had a more favorable safety profile compared to doxorubicin.
Doxorubicin.
Yeah.
That's a common chemo drug, but it has heart risks, right?
It does. Cardiotoxicity is a known side effect. So finding things that are effective and potentially safer is a huge goal.
And compound 5E seemed particularly promising, especially against breast cancer cells.
It really stood out, yes. In the lab tests against MCF7 breast cancer cells, 5E showed a greater cytotoxic effect. It killed more cells than doxorubicin did.
What did they find out about how 5E was working on those breast cancer cells?
They dug deeper into the mechanism. They saw morphological changes. The cells looked like they were dying.
Yeah.
They used specific stains, AOEB staining, to confirm apoptosis and necrosis were happening. Compound 5E also strongly inhibited the cell's ability to form colonies. And when they looked at the cell cycle, they found 5E caused the cells to get stuck, arresting them in the G1S phase, preventing them from dividing properly.
Stopping them from multiplying.
Right. And on top of that, it also increased the production of those damaging reactive oxygen species, the ROS, inside the cancer cells. So it's hitting them hard, stopping division and causing internal damage.
So similar strategies to the natural ones, maybe just better, more potent, more selective.
That seems to be the case, potentially. Improved potency and, importantly, that selectivity for cancer cells.
What about initial safety checks on 5E itself? Anything beyond cell lines?
They did do some preliminary in vivo work using zebrafish larvae. It's a common model organism used for rapid assessment of acute toxicity and potential cardiotoxicity early on in drug development.
Okay, an early glimpse at safety. And they also use computer modeling, right, in silico studies.
Precisely. Molecular docking studies. They used computer simulations to predict how well compound 5V might bind to key proteins involved in cancer.
Which proteins did they look at?
They focused on ones relevant to breast cancer, like the estrogen receptor, ER, epidermal growth factor receptor kinase, EFGR, the BRCA1 protein, and VEGFR2, which is involved in blood vessel growth.
And what did the simulations suggest about where 5E might be hitting?
The docking results showed strong binding interactions between 5E and both the estrogen receptor, ER, and the epidermal growth factor receptor, EFGR.
Suggesting those might be key targets.
Exactly. The model showed hydrogen bonds and hydrophobic interactions, suggesting 5V might exert its effect, at least partly, by interfering with the signaling pathways controlled by ER and EFGR, which are known drivers in many breast cancers. Interestingly, the binding to BRCA1 wasn't predicted to be as strong, maybe suggesting it's less likely to be a primary direct target.
It really sounds like these new derivatives like 5E are a big step. But the researchers still mention challenges, right? It's not a magic bullet yet.
Oh, absolutely. It's a crucial point. They emphasize the ongoing need for anti-cancer agents with better selectivity and fewer side effects. Doxorubicin's cardiotoxicity is a constant reminder of that need.
Yeah.
And translating promising lab results into drugs that are truly safe and effective in people is always a major hurdle in drug discovery. Target-based discovery has its own challenges too.
The articles also touched on other synthetic derivatives being developed. Any other quick examples?
Yes, the broader review mentioned a few others. Things like inhibitors designed to block the interaction between p53 and another protein called SNAIL that could be relevant for pancreatic cancer.
Okay.
Also, derivatives targeting the STAT3 pathway or cancer stem cells, which are involved in many types of cancer, and analogs of vitamin K3 that showed growth inhibition against multiple cancer cell lines. Often these studies report IC50 values, which give you an idea of how potent the compound is against specific cancer cells. It shows the breadth of the research.
Right, so let's try to wrap this up. After this deep dive into naphthoquinones and cancer, what are the main takeaways? What's the bottom line for you?
Well, I think the big picture is that naphthoquinones are a really fascinating and genuinely promising class of compounds for anti-cancer therapy. We're seeing potential from both the ones found directly in nature and the ones being cleverly designed in labs.
And they work in multiple ways.
Yes, that's key. They don't just rely on one mechanism. They generate ROS, they inhibit key enzymes like topoisomerase, they disrupt energy production, they modulate important regulators like p53.
And the research is pushing hard on creating better versions.
Definitely. The work on new derivatives, like that compound 5E, is all about improving efficacy and, crucially, improving the safety profile, making them tougher on cancer but gentler on the patient. That's the ongoing goal.
It's clear cancer treatment is incredibly complex, but naphthoquinones definitely seem to offer a really rich scene to mine for future therapies.
Couldn't agree more. The fact that nature provides starting points in chemistry allows us to refine them. It's a powerful combination. Which brings me to a final thought, maybe something for you to ponder.
Okay.
Given how widespread these compounds are naturally and our growing ability to synthesize smarter versions, what might the future of cancer treatment look like if we can truly harness these molecules more effectively, more selectively? Are there unexplored plants or microbes out there holding even better natural naphthoquinones? Or what clever chemical tweaks might unlock the next generation of really potent, safe anti-cancer drugs based on this structure? It's quite something to think about.
Thanks for tuning in to Bloom's Cast, brought to you by blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
Pharmacology.